Okay, can somebody confirm you can hear me?
I can hear you.
Thank you. Before we begin, are there any questions? All right, let me see. Chapter 2. Let's open up chapter 3 too. We'll start that shortly. All right, today is April 24th, and we're supposed to be on chapter 3. We're not there yet. There is a lab today. I'll talk about that in the lab time, lab 5. Any questions before we begin? All right, I'm not hearing anything, so let me begin. We have about 2 slides to cover in chapter 2, and then we'll begin chapter 3. We were talking about RNA last time. We need to discuss ATP, adenosine triphosphate. ATP is the primary energy source for all cellular activities. Whenever a cell needs energy for whatever purpose, it tends to get that energy from ATP. ATP has ribose, 5 carbon sugar, adenine, one of the um, nitrogen containing bases of DNA, and then 3 phosphate groups. Let me blow this up so we can see it better. This phosphate with the squiggly line is a high energy bond. The second phosphate is also a high energy bond. And if the cell needs energy, it can break this bond, and that will release a lot of energy that the cell can then use for its energy needs. If the cell still needs energy, it can then break that phosphate bond and get more energy. Any question about any of that? Whenever the cell needs energy, like I said, it takes it generally from ATP. So ATP is called the um, cellular energy of cells, meaning the energy of cells, because it's the currency that cells use to for their energy needs. Currency is something like money, and when we need something, we go and exchange money for what we need, what we buy. Well, the cells need energy, they get it from ATP. ATP is made by dehydration synthesis. You can take ADP, adenosine diphosphate, shown here, adenine with 2 phosphate groups attached, add an inorganic phosphate group and energy, and then this reaction can go that way, where water will come out and ATP will be made in dehydration synthesis. And then you can break ATP by adding water, and that will break off this third phosphate group, releasing a lot of energy, which some of that energy can be used for the cell's need, and that will make ADP, adenosine diphosphate, break off the phosphate group and release the energy. All right. Any question about any of that? So let's briefly talk about hydrogen bonds. What molecules have we discussed that have hydrogen bonds that give the molecule important properties? Anyone? What molecules have we discussed that have hydrogen bonds? that give the molecule important properties?
Water.
Yeah, water. That's one. Anything else? How about DNA? We have DNA, the two strands connected by the two strands of DNA are connected not by chemical covalent bonds, by what type of bonds? This group is not a very good group of contributing. You gotta,
Would it be the same situation, hydrogen bonds, or
yeah, hydrogen bonds are what's holding DNA together. So these are two important molecules that we've discussed that the hydrogen bonds give these molecule important properties. The hydrogen bonds hold two strands of DNA together, and the hydrogen bonds are what holds the water molecules together, so we have liquid water. What would happen to these molecules if hydrogen bonds were to all disappear?
Water splitting?
Yeah, you wouldn't have liquid water, and without liquid water, you couldn't have life. The DNA molecules would be single-stranded, so you wouldn't have double-strand DNA. And then there's other molecules that we have, like proteins and, I don't know, RNA and carbohydrates. They do form hydrogen bonds, and you would not have life if all the hydrogen bonds were to simply disappear in the universe. So it's just showing you that hydrogen bonds are very important for life. Boy, this is happening to this PowerPoint too. Because I'm working online, Microsoft every once in a while tries to verify my account to make sure I can use their Microsoft products, and I'm online, so they can't verify that I'm a Clark employee. So um, I tried straightening that out earlier, and it didn't work, so I'll have to do something else. But I'll have to get this taken care of before April 29th, or I won't be able to access any Microsoft programs. All right. Any questions about Chapter 2? If not, let's begin Chapter 3. Observing microorganisms. The major goals are shown in this slide and a rough outline. Always know the terms for each chapter. And really, you really need to know all the objectives, and you can find the objectives by looking on my study guide. Let me see if I can find it. There we go. So look at the study guide in the very start. I'll have the objectives. Chapter 3 objectives for observing microorganisms through a microscope. All right, let me go out of here. See if that'll close it. Did. And I can make Chapter 2 available to you while I'm here. All right. So to understand the sizes of organisms and the properties of microscopes, understand the major differences between a light microscope and an electron microscope. 3, understand why staining improves resolution. And know the 3 types of stains: a simple stain, a differential stain, and special stains. And then 3B, understand the Gram stain, its procedure, and the basis of its differential staining. Any questions about what we're going to be covering? So in Chapter 3, we're going to be using the metric system. This is a system for measuring units or using units for measurement. It is expected knowledge you will understand the metric system, such as 1 millimeter equals 1,000. Excuse me, 1 meter equals 1,000 millimeters. You should know most of this table below. Let me see if I can blow that up. You should know that 1 kilometer equals a 1,000 meters. Kilo equals 1,000. 1 meter is the basic unit for measurement of length in the unit. You don't need to know a decimeter, but you can if you want. A centimeter, you should know, is 1/100th of a meter. A millimeter, you should know that, is 1/1000th of a meter. And a millimeter is the smallest unit of measurement on a ruler. You should know a micrometer, that's 1/1,000,000th, or 1 out of 10 to the negative 6, or 0.000005 zeros, and then 1. This is the size we measure under the microscope. You should know a nanometer. That's 1/100000th of a meter, or 1 times 10 to the negative 9th meters. This is the size we measure under the electron microscope. You don't need to know a picometer. That's 1 times 10 to the 12th meters. And then you should know the metric system uses all the units in a base 10. So if you were to know the metric, a decimeter is 1/10th of a meter, a centimeter is 1/10th of a decimeter, or 1/100th of a meter, etc. And they're just easier to use, the metric system, because everything's based on a unit of 10. Switching from metric to US equivalents is difficult, and I've got it on the table here, but you do not need to know how to switch from metric to English and English to metric. Like about 0.6 miles equals 1 kilometer. You don't need to know that. You just need to know how to work in metric for this chapter, and really for the class. You need to know how to use metrics. Any question about any of that? Now, for the other units in metric, like meter is for measuring length, the liter is for measuring volume. You just use 1 liter, and then you use the same prefix, kilo for kiloliter, and centi for centiliter. So use the same prefix. And then for weight or mass, we use a gram as the basic unit. And once again, you use same prefix. We have a kilogram for 1,000 grams, and then a centigram for a 1/100th of a gram. Any question about any of that? You need to use metric for 2 reasons, and that is science and medicine use the metric system. And then this map is showing you all the countries in the world that do not use the metric system. And if you notice, there's only 3: United States, Liberia, and what is that? Siam? No. Yeah, I think it's Siam or Thailand. And 2 of these 3 are actually switching over to using the metric. The United States is actually switching over to using metric. If you notice on cars, it'll show you the miles per hour, and then usually above it in smaller lettering, it'll have kilometers per hour. And a lot of the road signs, it'll have miles per hour, and above it in smaller units, it'll have kilometers per hour. So the United States is slowly switching over to use metric, and I think that was actually started by Thomas Jefferson, stated that the United States will begin switching over to using metric. And as you can tell, we have been switching for a very long time and very slowly. We are mostly using the US equivalent, meaning the English system. Any question about any of that? All the other countries in the world use the metric system. And like I said, 2 of these 3 that don't are switching over to using the metric system. You should know the sizes of microorganisms. A bacteria cell is measured in micrometers. Eukaryotic cells can be measured in micrometers, but a eukaryote cell is about 10 times the size of a prokaryote cell. And this is about a range, the average range of a cell, like a chicken yolk cell, that's the largest cell you've ever seen, is larger than 500 micrometers. And what the heck would that be? The chicken yolk would be over a centimeter, maybe about 2 to 5 centimeters in length. The average prokaryote cell is about 1 to 2 micrometers. 10 micrometers is the length of the longest, probably prokaryote cell, but the diameter would be closer to 1 to 2, the average prokaryote cell. You can get them smaller too. I think the smallest viable cell is down to about 0.4 micrometers, so 0.4 micrometers. And they have smaller cells down to about 0.02 micrometers, but they think those are non-viable. And then the virus is about 1/10 the size of a prokaryote cell, ranging in diameter from 0.02 micrometers to 0.44 micrometers, meaning the largest virus reaches the size of the smallest prokaryote cell in its diameter. The longest virus is Ebola. It reaches about 1 micrometers long, but it's smaller in its length. And so the largest virus is actually in its diameter about 0.44 micrometers, which is megavirus. Any question about any of that? I blow this slide up. So once again, you should know this slide, that a human about the largest thing you can see. It's not entirely true. An elephant is larger, and then a whale, of course, would be much larger. But you can see a human with your eye. You can see the chicken yolk egg cell, the largest cell that you've ever seen. The largest cell on the planet at the moment is actually the yolk of the ostrich egg, but not too many people have seen that. I once had a student who saw the yolk of a turkey egg, but not too many students have seen that either. I've never even seen the yolk of a turkey egg. Frog egg, that's about the smallest cell that our human eyes can see. We can see a little bit smaller than that. And then we have the light microscope. Oops. Which sees things smaller than a frog egg. You can use a light microscope to see a frog egg too. Eukaryote cell, the average cell. The average prokaryote cell, smaller than a eukaryote cell. Viruses, smaller than a prokaryote cell. Large molecules, like large proteins, smaller than a virus. Small molecules like water, smaller than a large protein. And then atoms, smaller than a small molecule like water. And for seeing these small things, viruses down to atoms, you really need an electron microscope. We won't talk about anything smaller than an atom in this class. Any question about any of that? All right, so with the microscope, you need to know these parts of the microscope. We're going to be talking about a compound microscope. These are the microscopes we have in the lab, and a compound microscope has 2 or more lenses that magnify. The total magnification will be the magnification of the ocular lens 2 times the magnification of the objective lens. The typical microscopes in our lab have a 4 objective lens, a 4x objective lens, a 10x objective lens, a 40x objective lens, and a 100x objective lens. And I prefer these names because they're not confusing. You can call the 4x lens the scanning objective lens, the 10x lens, the low power objective lens, the 40x lens, the high power lens, and the 100x lens, the oil immersion lens. The trouble is that the low power lens is not the lowest, and the high power lens, objective lens, is not the highest. And some of you got confused in the lab and you got that wrong. So you should know the ocular lens, you should know the arm, oops, shown right there, for holding the microscope. Let's go back. You should know the specimen will be on the stage. We have a condenser. We have an illuminator, which is the lamp where the light comes from, and then the base. You should know these parts of the microscope, the body tube or the arm, which you hold it, the stage for holding the slide, the stage clips you should know for holding the slide in place and moving it around, the stage controls, is there. Stage control is there. The illuminator, the lamp, the coarse focusing knob, the big one here for moving the stage up and down at a faster rate, the fine focusing knob for finely focusing the object by gradually and slightly moving the stage up or down. You should know the on and off switch right there, and then the rheostat or the light intensity dial switch. You should also know, it's not shown here, you should know the iris diaphragm, which, oh, there it is. Sorry, it was there. It's here, right below the condenser. And like I said, sometimes the iris diaphragm will be above the condenser, sometimes it's below the condenser. You should also know a simple microscope, such as one shown here. This is the microscope that Antony van Leeuwenhoek made. A simple microscope only has one lens for magnifying the specimen shown here. And it is really, a simple microscope is only really a super duper magnifying glass because it has only one lens. You should know the resolution. That's the ability of the lens to distinguish 2 points as 2 separate and independent points. So a microscope that has a resolving power of 0.2 micrometers can distinguish 2 points that are separated by 0.2 micrometers or greater apart. If ever you have the 2 points closer than 0.2 micrometers, you cannot distinguish them as 2 separate independent points. The resolution of white light is about 0.2 micrometers. So this is the limit of a light microscope. The best resolution we can get using white light is 0.2 micrometers. And that is how far apart 2 points need to be before we can see them as 2 separate and independent points. The best compound light microscope has a total magnification of about 2,000 times. That will be 20x ocular times 100x oil immersion lens, or 100x objective lens for 2,000 magnification, and it will have the resolution of 0.2 micrometers. So if we were to ever make a microscope which had higher total magnification than 2,000 times, it would be empty magnification because it would be trying to magnify something greater than 0.2 micrometers, and the limit to white light is 0.2 micrometers, meaning you cannot magnify an object and separate it from another object by anything better than 0.2 micrometers apart. Because if you did, like I say, it will still be all blurry, and you would not be able to see the 2 points as distinguished from each other if they were closer together than 0.2 micrometers. So the resolution of white light is 0.2 micrometers, which is the total magnification of about 2,000 times. Anything greater than that will be empty magnification, so it doesn't really help you improve the resolution, doesn't help you see the object any clearer. Any questions about any of that? Now, this is the best compound light microscope that is in existence. At Clark College, to my knowledge, we don't have any microscopes with the 20x ocular lens. We only have microscopes with a 10x ocular lens. So Clark College does not have any of the best microscopes, and that's because they're kind of expensive, and we have a cheaper microscope that only magnifies 1,000 times. Any question about any of that? So here are 2 points which are separated by 0.2 micrometers or 200 nanometers, and you can see them when they're magnified as 2 separate and independent points. But these points are separated by less than 200 nanometers, and when we magnify that, we cannot distinguish them as separate and indistinguishable points. Like these 2 here look actually like 1 point. Any question about any of that? All right. Another property of light is it bends when it passes through a medium and goes into another medium. Like when light is passing through water and then goes out of the water and it's passing through the air. That makes the light bend at the switch in media. Why the pencil looks like it's bent, it's a really straight pencil, is just because of the refraction of light when the light leaves the water and enters the air, and vice versa when the light leaves the water and then, leaves the air and enters the water. Wherever the media changes, the light can bend, and we call that refraction, where the light bends. The refractive index is the ability of a medium to bend the light. Any question about any of that? The refraction is actually very important for light, like when a fish is in the water and a eye of the bird sees it, the bird can see the fish here, but it instinctively knows the fish isn't here, that the fish is really there, and it's looking like it's different because the refraction of light at the water to air interface, so that the fish is really here, but it's going to look like it's here because of the bending of light, and the fish will compensate instinctively. It sees the fish here, but it knows the fish is really down there. Any question about any of that? All right, the refraction of light is one of the reasons why we use oil with the oil immersion lens. And when we use oil, oil has the same refractive index as glass. And so when light leaves the glass and enters the oil, light will not refract or bend. And if you had the light leave the glass and enter the air, some of that light would bend. Because some of the light bends, some of the light will bend at a sharp enough angle that that ray of light will not be able to be captured by the objective lens. And so if we use oil, the light doesn't bend, we can capture more of the light by the objective lens, and that will improve clarity and resolution of the object or the specimen we are viewing. So to look at this in a little more detail, when we're using oil over here, all of the light coming through the lens will not refract. This one is not refracting, but it's at a steep enough angle, this ray of light, that it cannot be captured by the objective lens. But that one can be captured, that one can be captured, that one can be captured. When we are using air, this one can be captured because that light ray is actually not being bent. Not all the light is refracted, only some of it. This one is actually bent, coming out of the glass, entering the air, and it is captured by the objective lens. And you can note that the glass of the lens bends the light again, but it is captured. That one is coming out of the light and bent at such a steep angle that the refraction allows this ray to not be captured by the objective lens. So fewer rays of light can be captured by the objective lens when we're using air as opposed to using oil. And because the oil allows us to capture more light, we get better clarity and better resolution. And then this light, this ray here, is bent at such a steep angle when it leaves the glass that actually is bent back into the glass. So this one here, which was that one there. Anyways, the point is immersion oil is used to reduce the refraction of light, allowing us to capture more light, which improves the clarity and the resolution of the specimen we are viewing under the microscope. Any question about any of that? All right. Bright field illumination is what we normally use with our light microscope. In bright field illumination, the light leaves the lamp, goes through the condenser lens, passes through the specimen, and then will be caught by the objective lens, and then we can see the specimen with our eye. The specimen will be bright. The background will be very bright because we'll get the light from the lamp, and the specimen will be also bright. And that's why it's called bright field illumination. It is normally how we see an object with our microscopes in the lab. Any question about that? Dark field illumination is something different, where we have an opaque disc between the light source, the lamp, and the specimen. The only light that we can see is a ray of light coming out of the lamp at a steep angle, so it's not being blocked by the opaque disc. And then when this light ray comes through the specimen, some of the light is refracted by the specimen, and some of the light is refracted into the objective lens. So the only light we're getting from the lamp is actually light that is being refracted by the specimen into the objective lens. All other light at this steep angle, because it's blocked by the opaque disc at not the steep angle, all other light is either blocked by the opaque disc or is at such a steep angle that it is not captured by the objective lens. So this one will come through and will not be captured by the objective lens. Any question about that? That is why it's called dark field illumination, because the background of the slide is dark because it's receiving no light, because the light coming from the lamp is blocked by the opaque disc. The light that is hitting the slide is at such a steep angle, is not being captured by the objective lens. The specimen is also darker than the specimen under bright field illumination, and that's because it's receiving less light. It's only receiving light from the lamp at such a steep angle that most of the light is not going to be captured by the objective lens. The only light that's captured is the light that is refracted by the specimen, meaning bent by the specimen, into an angle that can be captured by the objective lens. Any question about dark field illumination? You do need to know the difference between dark field illumination and bright field illumination for the quiz. Here is an electron microscope, and you can see it's much bigger than an light microscope. An electron microscope uses a beam of electrons, not light, to see the specimen. And you're using the beam of electrons to magnify the specimen. Electrons have a shorter wavelength than light, allowing us to get greater resolution because the resolution limit is less than 0.2, micrometers. This allows the examination of smaller samples like viruses and internal cell structures. There are two main types of an electron microscope. There's actually 4 types, but there are 2 main types. That is a transmission electron microscope, shown in A, and a scanning electron microscope, shown in B. What do you notice between the 2 different specimens? It's actually 2 things you should notice.
One looks like it's showing the inner portion, one looks like it's showing the outside.
Yeah, the scanning electron microscope, we're seeing the surface of the cell in 3 dimensions. The transmission electron microscope is seeing one plane through the cell, meaning at 2 dimensions, and we're focusing in on inside the cell. You could focus in on a portion of the outside of the cell, meaning you go higher up on the cell, and you could see a portion of the surface of the cell, but you would not be able to see all of the surface because the surface is in 3 dimensions, and this is only looking at one plane through the cell. So the scanning electron microscope is seeing specimens in 2D. The transmission electron microscope is seeing the specimen in 2D. The scanning electron microscope is seeing the specimen in 3D. The transmission electron microscope is seeing one plane through the cell, and you can focus in on different, planes through the cell, meaning different areas of the cell. And the scanning electron microscope really only sees the surface of the cell in 3Ds. Any question about any of that? So this is a scanning electron microscope, seeing the crystal in 3 dimensions. And then this is a transmission electron microscope. I think this is the membrane of the chloroplast. And we're seeing just one plane through the cell, showing you the membrane of the chloroplast. There is 2 other forms of a microscope. You don't need to know them. You won't be examined on them. Scanning, tunneling microscope, using a metal probe to scan a specimen. The resolution of this one is 1/100th of the size of an atom. So we can see down to 1/100th the size of an atom. And here we're looking at a protein from E. coli. You can see it's seeing it in 3 dimensions. This, I think, can magnify the greatest, by the way. Scanning, tunneling microscope. We also have atomic force microscopy. You won't be examined on that. A 4th type of electron microscope, using a metal endowment probe inserted into the specimen, produces 3D images of the specimen. Here we're looking at the toxin from Clostridium perfringens with an atomic force microscopy. Any question about any of that? Here we're looking at two specimens using the light microscope. Specimen here where the cells are not stained, and there where the cells are stained. Which cells are easier to view? The cells on the left or the cells on the right?
Cells on the right.
Right.
The ones on the right. And that's simply because we have stained the cells. The cells are essentially transparent. There is very little difference between the, what's the word I'm trying to say, contrast of the cell and the background of the slide, meaning the glass of the slide or the water that the cell might be in, if this is in a wet mount. There's very little contrast between the cell and the background. If we stain the cells with a dye, there is much greater contrast between the cell and the background of the slide, which in this case would be just glass. And that is all that stains do. They increase the contrast between the cell and the background of the slide, which is usually just the glass of the slide. But it could be the glass in the water, like this looks like cells in a wet mount. So with bright field microscopes, that's the normal microscope we use in the lab, most microbes are colorless and have little contrast with the slide or the medium. To see them, you really need to stain them with a dye or a stain. Staining is just coloring the specimen or coloring the background to increase the visibility by increasing the contrast between the cell and the background of the slide. A smear is a preparation you make of a microbe, which is a thin film of a solution of microbes placed on a slide, ideally one layer of cell thick, but it can be more than one layer, and it could be, I guess, less than one layer. A smear is something you do with the microbes before you begin the staining process. So prior to staining, microbes in a smear are put on the slide, air dried, and then heat fixed to the slide to attach them. The heat fixing does tend to kill most, not all, but many bacteria cells. Usually the cells are fixed by air drying and then heat fixing by gentle warming. Any question about any of that? Let me end the show here. All right. Stains are a dye. So stains and dyes are two words which mean the same thing. And they are salts consisting of a positive and negative ion. The dye is in one of the ions. So the colored portion, the chromophore, is either on the positive ion or the negative ion. If the chromophore is a cation, meaning the positive part of the molecule, we call it a basic dye. If the chromophore, the colored part of the dye, is on the anion, the negative part of the molecule, then we call it an acidic dye. Any question about any of that?
Are you able to make that bigger?
Yes. Is that better? All right.
Thank you.
Now, let me get rid of that and I'll give you a little more room there. Bacteria have a slightly negative charge. Therefore, basic dyes are better for staining the cell because the positive basic dye will attach to the cell. The acidic dyes are actually repulsed from the cell because the chromophore has a negative charge and the cell has a slightly negative charge. The basic dyes are the most commonly used dyes in microbiology, like crystal violet, methylene blue, malachite green, and safranin are all basic dyes. Any question about any of that? Acidic dyes typically stain the background of the cell, not the microbes. Some acidic dyes are eosin, acid fuchsin, and nigrosin, and they stain the background of the cell, not the cell. I'll shrink that down just a little bit. Staining the background of the cell is called negative staining because the background is colored, not the cell itself. And this refers to the ancient Polaroid film where you had a negative and it looked inverse from the black and white photograph of the Polaroid film, where the light portions of the face will actually in the negative be darkly staining, and then the dark portions of the face, like the pupil of the eye, will be lightly staining. That's where the negative staining name comes from. Any question about that? Because the acid dye stains the background, when we use an acid dye, we tend to call it a negative stain. Here we're looking at cells, let me blow that up, that are stained with a basic stain. And as you can see, the cells are brightly colored. You can easily see the cell because it has a strong contrast with the background of the slide. You can also see the cells with an acidic dye that we call negative staining, and the acidic dye stains the background of the cell, but not the cell. But the contrast between the background and the cell is great enough that we can easily see the cell. Any question about simple staining and between basic dyes and acidic dyes? All right, as I stated, there are three main types of stains: a simple stain, a differential stain, and a special stain. A simple stain is just an aqueous or alcohol solution of a single dye. The primary purpose of a simple stain, meaning a simple single stain, is to increase the contrast between the cell and the background of the slide. This stain is applied to a fixed smear for a certain length of time. You then dump the stain off. It may be washed off, dried, and then examined. You may use a mordant. A mordant is a compound that simply intensifies the stain. Like in the Gram stain, we'll talk about Gram's iodine. It is a mordant. It actually physically combines in a complex with the the crystal violet, to make a bigger molecule than the crystal violet or the iodine alone. And the function of the mordant is to simply intensify the stain. The mordant is not a stain itself. It simply increases the affinity between the stain and the specimen, increasing the ability of us to see the specimen or intensifying the stain. A mordant may coat the specimen several times to enhance it, making it easier to see, like with flagella staining. The mordant, let me blow this up, coats the flagella many times, allowing us to see the flagella under the flagella stain. Now, blow it down now. All right, any questions about simple stains? If not, let's talk about differential stains. A differential stain is a stain that reacts differently with different kinds of microbes. Therefore, you can distinguish between two types of microbes. A differential stain usually uses two dyes, but this is not required. The most commonly used differential stains in microbiology are the Gram stain and the acid-fast stain. The Gram stain is a differential stain that we'll talk about in length in this lesson. It was developed in 1884 by Hans Christian Gram, so whenever you spell it, you should write it with a capital G because the stain is named after Dr. Gram, and we always use a capital G to name someone. And the Gram stain is actually named after the person who invented it. He used it as a differential stain to differentiate between the patient's cells and the bacteria that were causing the patient's infection. Microbiologists then started using it, and they realized it was very useful because it classified bacteria into two very large groups: the gram-positive cells, which stain a dark purple, and then the gram-negative cells, which stain a lighter pinkish color, separating most bacteria into two groups, the gram-positive and the gram-negative. And actually, you could say it specifies all bacteria into two groups. How you make the Gram stain is first you make a bacterial smear on a glass slide. You air dry it and heat fix it. That's to attach the bacteria to the glass of the slide, because otherwise the bacteria will wash off by the staining procedure. You then flood the slide with crystal violet. That's the primary stain for 60 seconds. You then wash that off with cold water. You then flood the slide with Gram's iodine, that's the mordant, for 60 seconds. You then wash that off with cold water. You then flood the slide with acetone ethanol, that's the decolorizing agent, only for about 5 seconds. You then wash it off immediately. You need to wash this one off immediately because the decolorizing agent, the trace of it left on the cells, will continue working, and you don't want it to continue working because you could, if you had enough time and enough acetone ethanol on the slide, you could pull out the crystal violet iodine complex from the gram-positive cells as well as from the gram-negative cells. You then flood the cell with safranin. It's the counter stain. A counter stain has a different color than the primary stain, and it's the second stain you use. You apply it for 1 to 2 minutes, and then you wash that off with cold water. You then blot it with bibulus paper. How you do that? We're going to pretend this is bibulus paper. You tear it off, fold it, pretend this is my slide here. You have it folded before you put the slide in here. And then you just press it down. You just blot it. You do not take the paper and then streak it across the slide. You don't take the slide and streak it across the paper either. You just blot it. That will remove enough of the water that you can view the specimen under the microscope, even if you're going to be using oil and the 100x oil immersion lens. Any question about any of that? All right, so in the Gram stain, this is showing you the procedure. We first stain with crystal violet. All the cells, gram-negative and gram-positive, stain a bluish purple color. We then apply the Gram's iodine. It will intensify the cells. That's not shown very well here, but actually the intensification is usually very slight. You will note that it will be a little more purple color than blue purple. And if you look really, really carefully, you may notice that the stain is a little darker, a little more intense. And that's because the Graham's iodine complexes with the crystal violet to make a larger molecule in the cells, but both the gram-negative and the gram-positive cells will be a purple color. You then apply the alcohol acetone wash, the decolorizing step, that will pull the crystal violet iodine complex from the gram-negative cells, but not from the gram-positive cells if you apply the decolorization step correctly. This is the differential step of the gram stain, because at this point you can notice a difference between the gram-negative and the gram-positive cells. You then apply safranin. It is a reddish or pinkish dye, and it will color the gram-negative cells pinkish. The gram-positive cells will continue to be a purple color. I don't know if I mentioned the decolorizing step will make the gram-negative cells clearish in color. And so the safranin just adds color to them so you can see them easier, because they'll have a deeper contrast with the between the cell and the background of this slide. Any question about any of that? All right. So before you begin the gram staining procedure, and actually before you stain on any cells, you always make the bacterial smear. So it is a step you have to do before you stain the cells. It is not part of the staining procedure. You make a bacterial smear on a glass slide, you air dry it and heat fix it to attach the bacteria to the glass. You then flood the cell with crystal violet, and if you did not heat fix or attach the cells to the glass, the crystal violet would actually lift most of the bacteria off the slide, and then when you dump that off, you lose most of the bacteria. So that's why you have to air dry and heat fix the slide. Anyways, you flood the crystal violet for 60 seconds. You then wash it off with cold water. At each step of the gram stain, there is a cold water wash. You don't need to state it when you're listing the steps of the gram stain. You don't need to put it in each step. You need to just state it one time, and after each step, you wash with each cold water. You can state it at the beginning or at the end. The water will remove the excess crystal violet from the background of the slide, but the crystal violet will not be removed from the peptidoglycan of the bacterial cell wall. The crystal violet is considered the primary stain, since it's the first stain we use, and it colors all cells the same. Any question about any of that? So after applying the crystal violet, both the gram-positive cells and the gram-negative cells will be a bluish purple color. You then apply iodine. Actually, I did state it up there. You wash it off with the cold water. You then apply iodine. Iodine is a mordant. It will complex with the crystal violet to make a larger molecule inside the cells. You apply the Graham's iodine for 60 seconds. You then dump it off and wash it with cold water. After applying the iodine, the cells will be a purple color, a little less blue purple, another more purple color. And you'd have to look very closely, but if you do, you might see its more intense color. You then use the colorizing agent. Usually we use acetone ethanol as the decolorizing agent, but you could use something else like ethanol acid as the decolorizing agent. You could also only use 95% ethanol. All of these are decolorizing agent. I think the video of the lab used 95% ethanol, and that's why you applied it for a longer period of time. 95% ethanol is not as strong, does not remove the crystal violet iodine complex as rapidly as acetone ethanol. When using acetone ethanol, you apply it only for about 5 seconds. If you only had 95% ethanol, you'd have to apply it for a longer time, and I think the video said for about 30 seconds. The acetone ethanol washes the crystal violet out of the gram-negative cells, but not out of the gram-positive cells, at least if you do this step correctly. The best way to do it is to hold up the slide so you can view it from the side, and then apply the decolorizing agent, watching the decolorizing agent, removing the purple stain from the slide. And once you see the purple stop streaking, you stop adding the decolorizing agent. And that'll be about 5 seconds. And then you wash that slide immediately with cold water to remove the trace elements of decolorizing agent from the slide, because it will continue to act. Any question about any of that? The timing of applying the decolorizing agent is important. You may decolorize not long enough, and it's also easy to decolorize too long. And you'll get problems if you do it not long enough or if you do it too long. And we'll talk about that in a little bit. So after the decolorizing agent, the color of the gram-positive cells is a purple color. The color of the gram-negative cells is colorless, meaning they're clear. You then flood the slide with safranin. It's this counter stain. You apply it for 1 to 2 minutes. Then pour it off, wash with cold water. Plot it dry, look at it under the microscope. The color of the gram-positive cells after applying the safranin will be a purple color. The color of the gram-negative cells will be a pink color. Safranin is a counter stain. It's applied for 1 to 2 minutes. Let me change that. All right, then you wash with cold water. And like I said, you only need to state when you're stating the gram stain that after each step, you wash with cold water. All right, the gram-positive cells are purple after the procedure. The gram-negative cells are pink after the procedure. Why? What's the basis of the differential staining? It's related to the differences in the cell wall structure. The gram-positive bacteria have a thick peptidoglycan layer. It's layer upon layer of peptidoglycan in these cells, at least 30 layers thick, up to several hundred layers thick. The gram-negative bacteria have a thin peptidoglycan layer in their cell walls. They also contain lipopolysaccharide layer as part of the cell wall. The thinner peptidoglycan layer is more than 1 layer thick. It's about 3 layers up to about 20 layers of peptidoglycan, but it's thinner, and the crystal violet iodine complex washes out of the thinner peptidoglycan layer. So we're looking at the gram-positive cell here. Let me load that up. Showing you yellow being the cell membrane, the purple being the cell wall, and it's mostly composed of peptidoglycan layer upon layer. It's only showing you 3 layers of peptidoglycan here, but like I said, it's something between 30 layers and several hundred layers of peptidoglycan. There are other molecules. We'll talk about those in a little bit. The gram-negative cell, on the other hand, has the same cell membrane shown in yellow, a much thinner peptidoglycan layer shown in purple, and then a lipopolysaccharide outside of the peptidoglycan layer. This layer here is the cell wall, which does contain the peptidoglycan layer and the lipopolysaccharide layer. Let me blow it up a little. On the gram-negative cell, we can see the cell membrane here. We have a thin peptidoglycan layer. It's only showing you one layer. Please remember it's between 3 to 20 layers of peptidoglycan. The book only tells you one layer or implies one layer and shows you only one layer. I think I got this from the picture from the book. But like I said, it's really between 3 layers of peptidoglycan to about 20 layers. And then showing you the lipopolysaccharide on the cell wall. That layer there is exactly the same or almost the same as the phospholipid bilayer of the cell membrane. This one is a little different. It does have phospholipid in it, probably the main molecule of the outer layer of the outer membrane of the cell wall. But it does have some different lipids in it, and then the old polysaccharide attached to that lipid. Anyways, the point is that this layer here is the cell wall, including the thin layer of peptidoglycan and what we call the outer membrane of the cell wall. The outer membrane of the cell wall does have phospholipid, the main molecule of the cell membrane, shown there and there. And it does have lipid A and the old polysaccharide attached to it, making the lipopolysaccharide. Any question about any of that? The important molecule of the gram-negative cell is this lipid A. Lipid A is an endotoxin. A toxin is something that is toxic to us. So lipid A is toxic to us. Any question about any of that? All right. So the basis of the gram staining is that crystal violet iodine complex forms in all cells, gram-positive and gram-negative. The crystal violet iodine complex is larger than the crystal violet or iodine alone. During the alcohol acetone wash, the crystal violet iodine complex is washed out of the cells because of their thinner peptidoglycan layer. The lipid polysaccharide layer is disrupted by the alcohol of the alcohol wash, and so the cells therefore appear colorless because there's no crystal violet iodine complex left in them. The gram-positive cells, the crystal violet iodine complex cannot be washed out of them because of the thicker peptidoglycan layer, and the cells therefore remain purple as long as we apply the decolorizing step correctly. So the picture of the gram stain will look something like that, with gram-positive rods, a gram-negative rod, a gram-negative spherical cell. What the heck is that called? Helical cell. I forgot the name. Anyways, and there's a gram-positive. There's a gram-positive cockeye. Another cockeye. There's a vibrio, a half of a spiral shown there. Any question about any of that? when we look at the Gram stain, we often see something like this. And how do you view that? What is that? Why are the cells much darker there than over here? And if you look, it's darker around this clump than further away or there. And how do you do that? When you see the stain, these are bad regions of the stain, and these are the good regions of the stain. So you make your determination in the good regions of the stain, not in the bad regions of the stain. These are actually crystal violet crystals, meaning crystal violet in the solution. The crystal violet can form a crystal where the crystal violet comes out of solution, grows into a crystal, and then that crystal was put on the slide, and that's actually a crystal of crystal violet. And as you can see, around the crystal, the cells have a darker color than further away from the crystal. It's like the crystal violet is coming out of the crystal and then staining the nearby cells darker than the cells further away, which is exactly what is happening. You'll note that the Gram stain will be a little difficult, especially when you're first learning it. Most consistent results are obtained when you perform the Gram stain on young growing bacteria. If your cells are greater than 2 days old, you should not use them to perform the Gram stain. You should take the cells, grow them up on a fresh petri dish or tube, and have them growing for less than 2 days, up to 2 days, and then use those freshly growing cells to make your smear for doing the Gram stain. The Gram stain is not universally applicable. Some bacteria stains poorly, if at all. So the results is not as clear-cut as theory suggests, and in the unknown project that may become apparent to you. The main point is when doing the Gram stain, make sure your cells are less than 2 days old. If they're older than 2 days old, don't use them for the Gram stain. All right, the structure of the Gram-positive and Gram-negative cells not only affects the staining of the cells, it also affects how the cell responds to antibiotics. The Gram-positive bacteria tend to be killed easily by penicillin G. Cephalosporin and many detergents also can kill Gram-positive cells fairly easily. The Gram-positive bacteria are more resistant to these antimicrobials, and the reason is the Gram-negative bacteria have an outer membrane on their cell wall. That outer membrane can help protect the cells, and for the penicillin G, for example, that outer membrane will not allow penicillin G to get through the outer membrane. That does not mean that we do not have antibiotics which cannot kill a Gram-negative cell. Simply penicillin G cannot. But ampicillin, which was the third member of the penicillin family, developed and then used, it is an antibiotic which can work on both Gram-negative and Gram-positive cells. So we do have antibiotics that can work on Gram-negative cells. Penicillin G is just one that cannot work on Gram-negative cells. Any question about any of that? All right. Special stains, you're not going to be examined on this, but you do need to know about special stains for your unknown project. Some of you may wish to perform the capsule stain for your unknown project. And the capsule stain is a special stain, staining the cell's capsule. Here we have a negative stain this may be a capsule stain using two different dyes to stain the cell, one to stain the background of the cell, and then the other to stain the cell. And you can see that the capsule is not being stained in the capsule stain. And so you can easily view the capsule as the region not being stained in this slide. Any question about that? And then another negative stain, which is a capsule stain for viewing the capsule, is simply applying India ink. India ink is this dark blackish color, and it stains the background of the cell, very dark, almost black color, or it is black color, whatever. And it does stain the cells lightly, like a grayish color, but it does not stain the cell's capsule. And so that's using India ink, a special stain, to view the cell's capsule. Any question about any of that? All right. If there's no questions, that's it for this lesson. Maybe I should just put this for my own reference to here. If there's no questions, I'll see you at 6:30 for the lab.